Hello students. So in this video we will discuss about Phyllotaxi, okay, or Phyllotaxi. What is this Phyllotaxi? The mode of arrangement, the mode of arrangement of leaves on the stem, on the stem or on the branch. So the mode of arrangement of leaves on the stem or on the branch is called Phyllotaxy or Phyllotaxy. Why they are arranged in a specific manner? Just to okay, uh, just to avoid overlapping and underlapping of the leaves, and so that every leaf will get sufficient amount of light. So to get exposed perfectly, they are arranged in a specific manner. And these methods of phyllotaxy, you can see among the plants which are of three types: alternate or spiral phyllotaxy, opposite phyllotaxy, and whorled or circular phyllotaxy. And only thing is that with the help of the diagrams, I hope you will be able to understand this phyllotaxy very easily. And we know that the place where the leaf originates is called node. And in this diagram, you can see that how many leaves are originating from this node? Only one leaf. And the next leaf is arranged alternate to it, not in the same direction or same line. So that's why this is called alternate phyllotaxy. And of course, when you examine this arrangement, if you take a stem, if you look at a stem, you'll find the first leaf here, second leaf here, third leaf here, fourth leaf here, like that. So if you draw a line, okay, you can see that the leaves are arranged spirally over the stem or on the branch. That's why this is called spiral phyllotaxy. And in opposite phyllotaxy, you can see here, this is the node, and at one node you will find what? Two leaves which are arranged opposite to each other. That's why this is called opposite phyllotaxy. Okay, the example for this is what? Calotropis and guava. Okay, I hope, right. Let us see the third one. And here this is the node, and at this node you will find more than two leaves. I drew four leaves here, and four leaves are coming at the same node, and they are arranged around the node like a whirl. That's why this is called whirled phyllotaxy or circular phyllotaxy. And the examples for this are Nerium and Alstonia. And I hope you are understanding the basic difference between these three. In alternate phyllotaxy, how many leaves are present at each node? One leaf. And in opposite phyllotaxy, how many leaves are present at each node? Two leaves. And in whorled or circular phyllotaxy, how many leaves you will find at each node? More than two leaves. So like that, you just remember that basic difference, and also you have to memorize these examples. Okay, China rose, mustard, sunflower are exhibiting alternate phyllotaxy. Calotropis and guava plant are exhibiting opposite phyllotaxy. Nerium and Alstonia are exhibiting whorled or circular phyllotaxy. Okay, that is about phyllotaxy, and the next aspect is leaf modifications. So now let us discuss about the modifications. Okay, already we know that what is modification? A permanent change in the structure of any organ to perform special function. So we already discussed some modifications in roots, some modifications in stems. Now we will discuss the modifications in leaves. Okay, in some plants like peas, sweet pea and garden pea. So in sweet pea, the entire leaf is modified into a tendril, entire leaf. So these leaf-like structures are not leaves; they are stipules. As leaf, entire leaf is modified into tendril to help the plant in climbing. So the photosynthetic function is taken up by the stipule and it got expanded and looks like what a leaf. And these leaf-like stipules are called foliar stipules. That's what you will find in the sweet pea, which is the scientific name is what, lathyrus. And in the similar way, you will find in garden pea, there is Pisum sativum. And in Pisum sativum, here also you will find foliar stipules. Foliar stipule, these are stipules. And the garden pea is having compound leaves. And you can see there is one rachis, and over the leaflets are present, but some of the leaflets are modified into tendrils. So which leaflets are modified? Not basal, the terminal leaflets. Terminal leaflets are modified into tendrils to help the plant climbing. That's what leaf tendrils. Leaf tendrils, of course, you will find in weak stemmed plants, and these tendrils will help the plant to climb. While they are climbing, they hold the support with the help of these tendrils. Okay, which are coiled, sensitive structures. Okay, now when we come to the spines, these are hard pointed structures, and these are seen among the plants growing in arid zones, means what, dry zones. So we discussed about Wapensia already, and in Wapensia, to minimize the rate of transpiration, the leaves are modified into spines. And of course, you know that the stem is became a flattened organ, and we call this flattened organ as what, phylloclade. So the main purpose of modification here is to reduce the rate of transpiration. But of course, as they are pointed structures, they also protect the plant from these animals, herbivores. So that is what the spines. And now, in some cases, the food will be stored up in the leaves. And the best example here is the onion and garlic. And here I drew the diagram of the onion. And you know that these are what the lamina of the leaf. And this is what leaf bases. So in onion, the food is stored in the leaf bases, and as they are arranged concentrically, so they appear in the form of a bulb-like structure. That's why we call this is what, onion bulb. So in onion bulb, what we are eating, the fleshy leaf bases that are storing the food materials. And in the case of garlic, of course, you will find some structures like this. Okay, and we call them what, garlic cloves, and these cloves are scale leaves. So the scale leaves are storing the food material in garlic, whereas leaf bases are storing food material in onion. Okay, now let us come to the fourth modification, that is what fill load. And what is this fill load actually? You can define the fill load as any part of the leaf other than lamina that expands and becomes a leaf-like structure and performs photosynthesis is called fill load. We know that the normal part of the leaf that performs photosynthesis is lamina. But any part other than lamina, if it becomes a leaf-like structure, expanded one, as you can see in the case of sweet pea and garden pea also, there are leaf-like structures. They are also fill loads actually. But generally the example that is given for the fill load is what Australian acacia. And here I drew one diagram. This is the leaf, which is actually bipinnately compound leaf. And this is the petiole. And this is the older leaf. And what has happened, the lamina portion is already okay, got detached, and only this expanded portion is left, which is petiole. And what you call this leaf like petiole that is performing photosynthesis fill load. Okay, so the example is Australian acacia. So now when we come to the trap leaves, so why they are called trap leaves? Because they trap the insects. I hope you are understanding. There are some plants which will trap insects and digest the proteins of that insect body as they are growing in okay, nitrogen deficient soils, which are called boggy soils. And we call these plants as what? Insectivorous plants. But insectivorous plants, they are actually autotrophs, and they show special mode of heterotrophism. Means what? They perform photosynthesis, but as in photosynthesis they prepare carbohydrates, but due to the non-availability of nitrates, they are unable to synthesize their own proteins. That's why they depend on insects when they are growing in such areas. If they grow in normal areas, of course they prepare proteins. Okay, and remember here some two examples we can take. One is Nepenthes and the other one is Dionia. Nepenthes is commonly called as pitcher plant, and Dionia is commonly called as Venus flytrap. And in I drew the diagram of Nepenthes here, and you can see this is the stem, and this Nepenthes is a climber, and you can see that this is the actually one leaf. And let us see the parts. This is the leaf base, and the leaf base, okay, the lower part of the leaf base, and this is upper part of the leaf base, and the lower part of the leaf base is modified into a phylloid. Is what it performs photosynthesis, and the upper part is modified into tendril. Okay, and this portion is what lamina. The lamina is modified into a pitcher-like structure, and at the apex you will find one lid, and it is immobile lid, and it is only for attraction. So this is lid, and this is the rim of the pitcher. Okay, so now whenever the insect enters inside, it falls inside, and of course here the enzymes will be there. There is digestive enzymes, and the insect will okay, will die whenever it drowns into that fluid. And this enzymatic fluid contains only proteases. That's why only proteins of the insect dead body will be absorbed and digested. And these absorbed proteins are again absorbed by this lower part. Okay, and is translocated to the rest of the plant body. And once the digestion is over, that means it has sucked everything. That means the enzymatic fluid along with the proteins is sucked, and the remnants of the dead body will remain there, and whenever it wants to, okay, throw it out, so with the help of the tendril it will become upside down, and as a result of which the dead body will be ejected out. Like that, you will find in trap leaves. So, in trap leaves, the lamina is modified to trap the insects. These are the different types of leaf modifications.